lab medicine we have to go through this today so you can understand the lab medicine. Regarding lab medicine, we'll go through electrolytes, we'll go through the urinalysis, we'll go through, well, a whole bunch of stuff. So, and then I'm going to give a case study at the end to try to tie it all together. Okay? The lecture objectives are here. We're going to go through embryology, we're going to do histology. Ultrastructure means at the electron microscopic level, so we can see some of the things I'm talking about. Then we're going to go into renal failure, acute and chronic renal failure. And some people this morning at Good Shepherd were asking me more to define acute renal failure. I mean, how do you get, what is acute renal failure? How do you define it, and what's it caused by, that sort of thing. So we'll touch on that today. A lot of renal disease is due to immune mechanisms. So we're going to cover those as well. And then we'll go over some signs and symptoms of post-strep AGM, and then a slide or two just on diabetes. You're going to have a lot more on diabetes when we get to endocrinology, but this is just as it pertains to the glomerulus. This is a light micrograph of the kidney showing the cortex and the medulla. Here's the junction between the cortex above and the medulla or medulla below, however you want to say it. In the cortex, you'll find the glomeruli. Also in the cortex, you're going to find ducts that are running as part of the glomerulus that you see here on this slide. You'll also have the collecting ducts. So the glomeruli are going to, and these convoluted tubules are going to drain into the collecting ducts. One collecting duct will receive dozens and dozens of distal convoluted tubules from nephrons. Down in the bottom here in the medulla, you're only going to see loops of Henle and collecting ducts and blood vessels. Now you're going to see a lot of blood vessels up at the top as well, because the glomerulus itself is really a blood vessel. Okay. So, you know, the weird thing about the kidneys or the GU system is that 1 in 10 people has a significant and serious congenital malformation of the GU system. I may have mentioned that in anatomy. 10%. So 3 people in here have a significant anomaly. You probably don't know it, but that's the truth. My wife, we didn't even know she had a non-functioning kidney until she was, in her 30s. So she had not only was it non-functioning, it wasn't there. A clump of something was there, but there was no functioning kidney there. So here she is with 1 kidney all her life. You've heard the expression ontogeny recapitulates phylogeny. Have you heard that? Raise your hand if you heard that. One freaking person in this room has heard that?
I took evolution, if that counts. That's where I heard it.
The rest of you have not heard that. I can't believe that. So what that means is that, you know what the phylums are. You know, kingdom, classes, families, phylums, right? So phylogeny is the arrangement of the species, right? Arrangement of the hierarchy of species. Ontogeny is the development of organs. So what that's saying is the development of organs recapitulates or is characterized by the ascendancy of the phyla. So if you look, I mean, a fish has some type of kidney. You do too. It's not the same. But as you go through the phyla from chordates to vertebrates to mammalians to humans to people from Arkansas, there is a development of a kidney throughout. Now, in the renal system, it's unusual because what happens is you start and stop the development of a kidney and finally get it right on the third time. Because there's so much starting, and wait, we're not going to have it that way, we're going to do it another way. And then you start to develop it, wait, we're not going to do it that way, we're going to do it this way. At each of these steps, you introduce the probability of error. And that's why you have 10% of people with a congenital malformation of the GU system. So, the most ancient kidneys, if you will, or primitive kidneys, would be the pronephros. The pronephros starts to develop in humans, but it never makes it to the point of filtering anything. You can see where it's starting to develop, but it never goes anywhere. Unlike primitive fishes, lampreys and whatnot, that's the kidney they started with and that's what they stuck with. All right, so you decided, okay, we're not going to do this. We're going to do the mesonephric kidney. So the mesonephric kidney starts to develop. And actually that does become functional in humans for a very short period of time. But then it starts to degenerate. And then long about week 5, and I'll explain this week 5 here in just a second, you start to develop the definitive kidney, which is the metanephric kidney. This metanephric kidney is going to start becoming functional at about the end of the first trimester. It's going to start producing urine by the end of the first trimester. All right. Now, for a little embryology here, we're going to cover this in much more detail when we get to the placenta in OBGYN. But, you know, when you are conceived, the first week, you are developing into just essentially a solid ball. And you make your way into the uterus where you implant. So by the end of the first week, you've implanted into the uterus. By the end of the second week, this is what you look like here. That ball is turned into, or the solid ball is turned into a hollow ball, really, with a 2-layer primordium here, separated on the top by what's going to become the amnion, and what's on the bottom is going to become the yolk sac. All this stuff out in here, this pinkish rose-colored stuff, is going to become the placenta. So by the second week, this is what you look like. You're 2 layers, you're 2 cell layers thick. You're a plate with 2 cell layers thick. You got an epiblast and a hypoblast. That's all you got going for you right now. Okay? Wait 1 more week, and then what happens? And now up here at the top, there's your 2 weeks. There's your 2 layers, amnion, yolk sac relationship. Remember, this is embedded in the uterus now. Along the 3rd week here, what happens is that flat plate, you see there's the flat plate right there. That oval plate has a streak in the middle of it, and you get these cells that migrate from the top into that streak so that they situate themselves between the epiblast and the hypoblast. So now you have 3 layers. At the end of the 3rd week, you have 3 layers. An epiblast is going to become skin and nervous system. The hypoblast, which is going to become the gut lining. And the mesoderm, which is in between, which is going to become everything else. Your muscles, your bones, all that other crap. Okay? So now you got 3. So it's really cool how this works out. You know, the first week you're just a ball. Second week you're 2 layers thick. The 3rd week you're 3 layers thick. Perfect. By the end of the 4th week, or into the 4th week and the 5th week, So you're still a flat plate here by the end of the third week. You're still a flat plate right now, right? Not many people look like that. So what happens in the fourth week, in the fifth week, is you start folding. This plate folds. So that it will fold from laterally, and it will fold from caudal, the cephalad to caudal, to form this thing right here. It could be a bird, it could be a shark, it could be a human. Okay? Depends on whose uterus you happen to be in. So if you look at here around the fifth week, this is what we look like. That yellow stuff there is the primitive hypoblast. That forms the gut lining. That will give rise to the liver, the pancreas, the gallbladder, the GI tract, right? The thyroid, the lungs. You got the primitive epiblast is going to be, as I said, the skin and the nervous system. And everything in between is everything else. Everything else includes the urogenital system. Now right here, you see those two little dots? Those two little dots consist of what's called mesenchyme. It's the middle tissue, right? Here are the two little dots right here, these circles. They form a ridge down the back of the inside of the abdomen. They form a ridge. You can see it. Here's your gut. Here's the this is going to be the intraperitoneal cavity here. They form a ridge right here on your back. The back of the abdominal wall. I don't mean your back, the back of the abdominal wall. It's called the urogenital ridge. You can see it over here. The urogenital ridge extends all the way from the cervical region all the way down to the caudal region of the embryo. Here's the thing you should already be picking up on. If I'm calling this the urogenital ridge, odds are good that that's going to give rise to the kidneys, right? What are the kidneys in terms of their relationship to the peritoneum? They're retroperitoneum. Why? Because they develop behind the posterior parietal peritoneum. They never were invested like the gut tube. Okay? All right, so there we are about the fifth week here. If we look at it like this, here's the pronephros way up here at the cervical region. It started to develop and then just petered out. Here's the mesonephros, the dark colored stuff here. It's made up of two parts. One part is a duct. A duct forms called the mesonephric duct. The other part that forms is the mesoderm around the area in the urogenital ridge. That mesoderm formed into little vesicles. The little vesicles started elongating. One end of the elongated vesicle joined the duct. The other one flared out and surrounded a capillary. That's a nephron. So for a short period, you had functioning mesonephric kidneys running along that urogenital ridge. And so if you look at this thing right here, this is exactly what Professor Polk has drawn on the board. There's the glomerular tuft, convoluted tubules, loop of Henle, convoluted collecting duct, right there. That's what you got. The collecting duct in the mesonephric kidney is the mesonephric duct. There's only one. There's only one collecting duct on each side in the mesonephric kidney. So you're rather limited about how many nephrons you're going to be able to build, right? Doesn't matter because this is all going to degenerate anyway. I got a question for you. if you had an ectopic kidney, well, let me save that question. Let me save that question. I'm going to give it to you in about another 3 minutes. Way down here at the bottom of the mesonephric duct, a diverticulum develops called the ureteric bud. You see that little thing there, ureteric bud. You can see it better in this illustration right here. The ureteric bud is a diverticulum off of the mesonephric duct. You see it? Everybody see it? Here's the mesonephric duct up here. Here's the ureteric bud. That ureteric bud is a diverticulum, and of course, as it grows, like all diverticuli, it forms the thyroid, the lungs, and everything else. As it grows out, it starts branching, and then you have all these branches, right? All these branches. Well, that's the same thing that happens here. This thing branches and branches and branches and branches and branches until you've got millions of collecting ducts that all drain into the minor calyces, that drain into the major calyces, that drain into the papilla, that drain into the ureter. So the ureter comes from the ureteric bud. The collecting system of the kidney is from the ureteric bud, which is really a branch of, or a diverticulum off of the mesonephric duct. So you can really think that the collecting system of the kidney is mesonephros in origin. Now, as these things grow, they're surrounded by mesoderm. Remember, the whole urogenital ridge is mesoderm. Similar to the mesonephros in forming the nephrons, they form here as well. So you get all of these. Now here's the mesonephros, mesonephric duct, ureteric bud. Here's the mesoderm here. Within that mesoderm, you get the formation of the nephrons. So the tubules themselves, and the glomerulus here, the capsule, its origin is mesenchyme, or mesoderm. Right? So they come from two different places. When you're born, these things, well, let's say, these things become functional, as I said, about the end of the first trimester, right? You make nephrons consistently throughout the rest of the pregnancy. So that when you're born, you have about 800,000 nephrons or so in each kidney. And for the first several months of your life, a couple of years actually, you make more. So that by the time you're an adult, you wind up with about 1 million nephrons in each kidney. That's pretty cool, huh? That's pretty cool. Any questions about this? Well, this is what you end up with. You have a collecting duct. Here's the papillae, minor calyx here. All these nephrons are draining into a collecting duct that is, for all intents and purposes, mesonephros in origin. And then you have the nephron itself. There's a transition between the cortex and the medulla. You can see in this light micrograph over here, a glomerulus there, there's another one there, there's a vessel there. You can see all these ducts cut in different cross-sectional planes. So here's sort of a summary, that first bullet point. But the second bullet point is the kidneys develop with their hilum facing anteriorly and in the pelvis. That's where the metanephric kidney is, right? But that's not where your kidneys are. Your kidneys are up here and they're rotated 90 degrees so that they actually face medially. That's what has to happen. Your kidneys, as you develop, ascend out of the pelvis and rotate medially. And I explained during anatomy, what happens is, as they ascend, new blood vessels will grow into them, and the lower ones will die off, so that you wind up with the renal arteries up here. But one of the anomalies is a pelvic blood supply. That's why they carried it with them. In some people, they will have pelvic kidneys because they never ascended. If you're going to find, and here's the question I was going to ask you earlier, if you're going to find an ectopic kidney, where do you think you find it? Say it again. No, if you're going to find an ectopic kidney, because right now, those would be your metanephric kidneys, right? So an ectopic kidney, what do you think an ectopic kidney would be? It would be a persistent mesonephric kidney. An ectopic kidney would be, wait a minute, the mesonephric kidney never went away. It stayed. Where would it be?
Higher.
Right, it would be above wherever the metanephric kidney was. So in some people, when you get when you do imaging on them, you're going to find either with a CT or with even a plain film of the abdomen, you'll see the kidneys. You might see another kidney above here, above it. They're always smaller. They're always smaller. Sometimes they're actually functional. That would be a functional mesonephric kidney. Okay. Now, that happens pretty early on, by the first trimester as well. Now let's talk about urine. If the kidneys are starting to function, where does all that pee-pee go? Okay? Because the embryo is sitting in your uterus within a chorion and amnion. He's peeing amniotic fluid. Amniotic fluid is pee. Not all pee, but it's mostly pee. Well, if the embryo and fetus is going to be peeing several hundred cc's a day, it's not going to take too many days before mom explodes. What happens to all that urine that's created by the embryo and fetus? You drink it. You drink it. You drink the amniotic fluid. It gets absorbed in the GI tract. Now it's into circulation. It goes to the placenta. The placenta gets rid of it. That's how you get rid of the urine you make every day as a fetus. By the end of the first trimester, you're not an embryo anymore, you're a fetus. Okay, so what happens if you have esophageal, some congenital development of the esophagus where it doesn't form, esophageal atresia, or you have, when you start out with your mouth, there's a membrane over it. What if that membrane persists and you can't drink your amnion? That's what's called polyhydramnios. What happens if your kidneys fail to develop? You don't make enough amniotic fluid. That's oligohydramnios. So when you get a fetal ultrasound, the tech will always make a comment about the volume of amniotic fluid. You mothers out there, do you remember that? They always make a comment about the volume of the amniotic fluid to make sure there's not an issue here. Any questions about that? All right. Histology of the kidneys. These little guys, they make up such a small amount of your body weight, but they, as I told you in anatomy, they take up 25% of your entire cardiac output. That's tremendous. That's tremendous. Where does the other 75% go?
Everywhere else.
Well, yeah. Break it down by 25%. Where does 25% go?
Brain.
Your brain. Where does 25% go?
GI tract.
GI. Where does the last 25% go? Everywhere else. Right, okay. The kidneys function, they have a whole host of, we have a guest today. Would you like to introduce the guest?
Sure. This is my boyfriend, he's a med student, and he just wants to sit in on one of our lectures.
All right.
What's his name?
I'm Brandon.
Okay. So the function of the kidneys, numerous functions that you got to cover over the next 2 weeks here. The endocrine function, we've already talked about the endocrine function. What's the endocrine function of the, what does a person with chronic renal failure always have? Anemia. Why do they always have anemia? Erythropoietinogen or erythropoietin, right? That's an endocrine effect, right? This thing right here that she just talked about this morning, renin, that's an endocrine effect, right? Regulates water and salts. We're going to talk about that today and tomorrow extensively. Maintains the pH. We already know that, right? We already know that. What gets rid of CO2? The lungs. What gets rid of, what manages, if the lungs are not doing the right thing, what's the counter mechanism? It's the kidneys, with getting rid of bicarb or retaining hydrogen ion, right? Okay, and excretion. Well, we got to get rid of all sorts of excretory products, the end products of metabolism. We got to get rid of that. It's like asparagus. You eat asparagus, and the first time you pee, it's like, oh my God, what is that smell? Right? I can't remember what the name of the compound is. Anybody know what the name of the compound is? What? Asparagine? I don't know what it's called. It's called skunk is what it's called. I did a study on myself one time. I timed myself at how fast that smell could enter your urine. You know, it's less than 5 minutes. I would eat an asparagus and I would make myself go pee. And it's like, there you go. All right. Tom, figure it out your own self. If you can make 5 minutes, I want to hear about it. Okay? Now, the big structure we're going to pay attention to today is the glomerulus, and you've already obviously talked about the glomerulus. The glomerulus is consists of these anastomosing capillaries. There's an afferent arteriole and efferent arteriole. Did Professor Paul go over that earlier? Good. The part of the collecting system here forms a capsule. That capsule, though, has a couple of different layers here. This is actually not drawn correctly. This should be actually all the way over. There's no space leaving that there. It's connected. Now, that connection means that right here, there's a parietal layer, and right here, there's a visceral layer, and they're contiguous. That forms the capsule. It's not open, it's sealed off. The space in between there is called Bowman space. That's where the urine is going to go. The urine is going to leave the vascular space and enter Bowman space. Before we get to that, let's talk about the blood vessels. How do you get to the capillaries? Well, you've got the renal artery, and as soon as it enters the kidney, the 1 or 2 renal arteries will branch into interlobar arteries. So the interlobar arteries go between the papillae. They divide at the junction of the cortex and the medulla into the arcuate arteries. Off of the arcuate arteries are the interlobular arteries that go up like that into the cortex. And then off of the interlobular arteries, you'll have the afferent arterioles. There's the afferent arteriole feeding in to form the capillary tuft. The efferent arteriole is where it collects. The vascular that surrounds the nephron is called the vasa recta. And so if you filter urine, most of what you filter gets reabsorbed in the proximal convoluted tubules. If it gets reabsorbed, it's got to re-enter the vasculature, right? So that's why you have a heavy blood supply around the nephron itself. And then they're going to reorganize into the interlobular vein, arcular vein, interlobar vein, renal veins. Pretty easy. Now, this is what it looks like. Here's the afferent arteriole right here. Right there before it goes into the breaks into a capillary though, there's a series of cells here that are distinct. That's called the JG apparatus. You talked about that this morning, juxtaglomerular apparatus. That's part of the afferent arteriole, the JG cells. Then here is the visceral layer of the capsule. Surrounds the capillaries. If you'll notice, they have interdigitations. So the interdigitations are like this on your fingers. So the urine, when it comes, or the plasma, when it's filtered and it enters Bowman space, it goes between these slits. We'll go into these slits with a little bit more detail in a second. But the urine enters Bowman space and then enters the convoluted tubule here. So this is the parietal layer of it here. This is the visceral layer, and you see they're continuous there. Now, as you got the proximal convoluted tubules, the loops of Henle, the ascending loop of Henle, thick limb, and then you've got the distal convoluted tubule up here. That distal convoluted tubule passes right by the afferent arteriole, right there. And there's a series of cuboid cells that are distinct in the distal convoluted tubule called the macula densa. So you talking about the macula densa? Okay, we'll got a couple of things to say about it, and then we'll move on. Any questions about this? All right. Here's the thing I want to say about the macula densa. It relates in the JG apparatus. It relates to the concentration of salt, sodium chloride in particular. Okay? Did she go over this? So I don't really need to mess with this. If sodium chloride levels fall, it's detected by the apparatus here, causing an increase in blood flow in the afferent arteriole. That increases the hydrostatic pressure inside the glomerulus, causes more urine formation, which causes more renin production. Actually, when I say it causes more urine formation, it's the increase in hydrostatic pressure that causes more renin production. Renin, the ultimate result of renin is to reabsorb sodium and water. Okay? The opposite is in the last paragraph. All right. So if you look at, so this is what I want you to know. If you look at the layers of the glomerulus here, what do you have to go through to form urine? Well, first you have to be able to make it through the fenestrated endothelium. So if you look at the capillary here, the capillary is fenestrated, has little holes in it. I think I've probably mentioned this before. These holes are actually pretty small, around 100 microns, nanometers in width or diameter. If you compare that to a red blood cell, which is 7 microns or 7,000 nanometers, you're not going to get a red blood cell typically going through the fenestra. Now, if you get through the fenestra, then the next thing you run into is the GBM, the glomerular basement membrane. The GBM, you see, is right there. It's secreted by the podocytes. Well, let's not worry about where the GBM secreted. It's mostly secreted by the endothelium. The podocytes, the foot processes of the podocytes are embedded in it, and it's really one structure. Let's go with it that way. So we've got the GBM, then we've got secondary foot processes. If we look at these things called podocytes, is it hot as hell in here or what?
Yeah.
Can we turn a fan on, or is that the way you do it here? By the way, I sent an email over to the head of facilities day. We copied his boss in the administration. I said that the temperature inside this building is not acceptable. And she actually told me to send him this email, so she's gonna eat his ass tomorrow. So, just let you know. Let me turn this thing on.
This is done.
Everybody's smarter than me.
Yeah, I just
We open the
have a
doors.
vision.
This 15 degrees cold out there. Okay. Okay. These are the podocytes. They have primary foot processes and secondary foot processes. It's the secondary foot processes that interdigitate. Between the foot processes, though, there is a, the diaphragm between the foot processes is a diaphragm. It's called slit diaphragm. Okay? The foot processes are separated by a few nanometers, but then you've got a diaphragm on top of that. This diaphragm is made up of a couple of things. One is heparan sulfate. The other thing is type 4 collagen. I don't know if I say that up there or not. The diaphragm is made up of heparan sulfate and type 4 collagen. So if you look between these foot processes here, you can't see it in this image here, there would be a membrane between the foot processes. So in order to get urine, you've got to be able to pass through the fenestra, you've got to be able to pass through the GBM, you've got to be able to pass through the secondary foot processes, you've got to be able to pass through the slit diaphragms. And then you're involved in space. So that's a lot of stuff to get through to be filtered. So here's what it looks like under a scanning electron micrograph. Between the secondary foot processes, this is the slit diaphragm here. Now one of the proteins in the slit diaphragm is nephrin. The absence of nephrin due to a congenital defect in the gene is what forms nephrotic syndrome, or what results in nephrotic syndrome. What's the chief thing about nephrotic syndrome? You pee protein. You swell because you pee protein. The oncotic pressure changes, you pee protein, you swell. You pee protein because it gets out through the slit diaphragm, or the absence, or defect in the slit diaphragm. Okay? So in this cartoon here, what you have to understand is all these illustrations of the vascular tuft here, they always show it as there's a lot of space in here, right? There actually is no space in there. That area is occupied by a cell called the mesangial cells, mesangia, that secretes a substance that actually forms sort of a gelatin that the mechanism, the glomerular tuft, rests in. The thing about these mesangial cells is they have immunologic properties. If they're ticked off, they can divide, they can secrete more mesangium, they can make the tuft more gooey. They have phagocytic properties, they can go eat junk that winds up in the tuft. So they're not just static cells, they're very active cells. And then if you follow this, you ought to be able to identify things. Here's the vascular endothelium, here's the fenestra, there's the GBM, secondary foot processes, primary foot process, podocyte nucleus, mesangial cells, and so on. Right? It's on video.
That's on, right?
Oh, yeah.
I hope so.
We'll make sure of it.
Well, if it's not on, there's a summary slide that says everything I just said. Okay, so at the electron microscopic level, this is where you can really see things. This is a platelet right here. This is plasma. See all that flocculent stuff? They're plasma proteins. This is the endothelium here. You see a hole there, and there's a fenestra right there. This space here, that flocculent stuff, is the GBM. Here are the secondary foot processes right there. And you get up here, you look at some of these things, you can see a little line running from one foot process to the other. That's the slit diaphragm. So out here would be the mesangium. There's a mesangial cell right there. Kind of cool, huh? All right. Oh, any guesses to what that thing is right there? That's the nucleus of the endothelial cell. All right, and this is a cartoon. I took this and just I put it in here because I really like the way it looks, for no other purpose. Because you can see the foot processes really well. Okay. Here's the summary slide that contains everything I've told you since January. All of these numbers and letters here are on the legend in the next slide. You thought I was going to start naming all that crap, didn't you? No. It's all right there. Okay? So you can go through there. You should be able to recognize all of this stuff that we've gone through. Okay? So, summary, filtration is based on the fenestra, the slits, the diaphragm, all this other stuff. Because of their small size, water, electrolytes, ions, they go through pretty readily. But once you get to things with the size of albumin, or especially red blood cells, or proteins, immunoglobulins, and things like that, they don't go through there so well. It's based not only on the size of the molecule, but also on the charge. Things that are very anionic, like or negatively charged, like albumin, they are repellent. So not only do you have a size factor, you have a charge factor that influences filtration. Now, I'm saying influences filtration as a process. There are many things outside of size and charge that alter filtration, and we're going to go over some of those tomorrow, like the pressure of blood in the renal arteries, for example. Okay? All right. The tubules, the only thing I want to say about the tubules is it's the proximal convoluted tubules that do most of the work, and because they're the ones that are exposed to the highest concentrations of toxins, they're the ones where most of the damage is going to occur in the proximal convoluted tubules, the glomerulus and the proximal convoluted tubules. That's really all I want to say about that. Okay? I'll only take a break, and then we'll switch gears and move more into clinical application.